The fetal pig is one of the most common mammalian dissections. Due to its state as a fetus, its prenatal functions can be compared to that of adult mammals, and a comparison of anatomies can be critically observed. Please select a lesson, and then press click here to begin.
Digestive system.
In this lesson you will do the following. Follow the path food takes. Identify organs of the digestive system. Open the stomach and cecum. Examine villi under the microscope.
The digestive system of the fetal pig relies upon nutrients transferred from the mother's placenta through the umbilical cord. However, in later stages of fetal development, the fetal pig's alimentary canal is mature enough to function should birth occur. We shall now follow the alimentary canal pathway that food would take in a postnatal specimen. Press the scissors button on the toolbar. Cut away the external flaps for better observation. Excellent. Now examine features of the neck and locate one of the mandibular glands. Good job. The mandibular glands along with the parotid and sublingual glands make saliva for digestion. Find one of the parotid glands. Correct. Saliva enters the mouth through a network of ducts that lead into the mouth, and begins to break down food with its digestive enzymes. Now open the mouth, and press the magnifier button. Lift the tongue. Under the tongue are saliva ports that lead from the sublingual glands just below the floor of the mouth. The tongue itself is a group of muscles that are coated with papillae and taste buds. It pushes food to the teeth for grinding, and is also important in swallowing. After the teeth mash the food with saliva, the tongue forces a portion of it over the folding epiglottis, and into the esophagus to be swallowed. The esophagus is a flat muscular tube that lies dorsal to the trachea, which we removed in the respiratory lesson. Close the mouth, and press on the esophagus utilizes muscular contractions called peristalsis to move food into the stomach. In effect, it squeezes the food there, much like toothpaste from a tube. Lift the liver, and then find and press on the stomach. Press the scissors button. Cut around the perimeter of the stomach, and then open it up. The dark material inside is called meconium. It is made of bile, mucus, and shed digestive lining. If you clean up the meconium with the cotton swab, you should see the smooth inner walls of the stomach. The stomach uses enzymes and hydrochloric acid secreted from these walls to digest food. You should now see a sphincter at its cranial end called the cardiac valve. Press on it. Good job. The cardiac valve prevents acids from rising into the esophagus, which connects here. Strong muscles within the stomach walls knead the food until it becomes a soupy mixture called chyme. This chyme settles naturally at the caudal end of the stomach called the pylorus, where it waits for entry into the small intestine through the pyloric sphincter. Press on the pyloric sphincter. Excellent. The chyme then passes through the pyloric sphincter, and into the beginning of the small intestine, called the duodenum. Fold back the stomach, and press the magnifier button. Now press on the duodenum. Good. The duodenum is connected to the pancreas, by way of the pancreatic duct. Identify the pancreas by pressing on it. Correct. The pancreas releases digestive enzymes, which break down different types of food. One particular enzyme is lipase, which aids in the breakdown of fats and glycerol. The pancreas also releases carbonate ions that can neutralize the acids within chyme, so that it doesn't damage the intestines. Now locate the teardrop-shaped gallbladder on the underside of the liver. Excellent. The gallbladder stores a substance called bile from the liver. Bile contains bile salts that help promote the breakdown of fats along with lipase. It is also alkaline, so that it too helps reduce acidity in chyme. The color of bile is dark, and it is what darkens feces. Bile gets these pigments from recycled hemoglobin delivered from the ribbon-like spleen attached to the side of the stomach. A cystic duct transports the bile from the gallbladder, and merges it with bile from the hepatic duct of the liver. Press on the cystic duct. Good. Together these ducts form the common bile duct, which also empties into the duodenum through a small sphincter at its end. Press on this duct. Correct. The combined mixture of neutralized chyme and enzymes then moves into the small intestine, also known as the jejunum ileum. Pull the jejunum ileum to the pig's right. Press the magnifier again. Nutrients are absorbed throughout the length of the jejunum ileum into tiny vessels that lie within the clear mesentery, holding everything together. This mesentery also prevents the intestines from tangling. Press on it. At the very end of the jejunum ileum is a protruding sac called the cecum, which is part of the cecocolic junction. Press on the cecum. Good. One possible reason for the formation of the cecum is to divert pressure away from the Allele Sphincter, which separates the large and small intestines. Press the scissors button and cut open the cecum. Next, press the magnifier button. Identify and press on the Allele Sphincter. The Allele Sphincter's job is to prevent the backflow of contents from the large intestine, or colon. Inside the large intestine, water is absorbed back into the body. Also, symbiotic bacteria break down undigestible materials here, to make vitamin K and sugars. Now press on the spiral colon. Good job. The spiral colon is coiled up with connective tissue. It exits dorsally into the ascending colon just under the pancreas. Press on the ascending colon. Good. Now continue by pressing on the descending colon. The descending colon then enters the pelvic cavity as the rectum, and exits the caudal end of the pig at the sphincter called the anus. Press the scalpel button. Cut a section of small intestine to view under the microscope, and then press the forceps button. Place the cross-section on the microscope slide, and then press the microscope button. As you focus the microscope, You should see the many villi protruding from the inner walls of the small intestine. Each individual villus absorbs nutrients into its capillaries, and as a group, they create a greater surface area to absorb nutrients. You have now completed this lesson. Press the arrow to continue.